What is sterilization?
To remove or to kill the complete microorganisms.
Complete removal. Okay, complete removal. This sterilization can be complete or can do for surface. Now, complete means you are doing the complete sterilization of culture vessel. Culture vessel. That means the vessel, if you want to do sterile, you will completely sterilize that culture vessel. Next, nutrient medium. Yesterday's class, we have seen now the composition of nutrient medium. So that nutrient medium should be completely sterilized. Why? So that no contamination will remain. The chemicals which you are using, that should be completely sterilized, as well as the instrument. Instrument can be your test tube, glass pipe, petri dish, whatever it can be. Okay, and suppose for this complete sterilization technique, what you will use? The instrument which you will use, the name of the instrument is autoclave. Autoclave is the name of the instrument. Suppose it is actually you have not seen this structure. This structure looks like this. Very large vessel like this. Autoclave. Okay. And here a machine is there which will measure the pressure. Okay. Now its working function same like pressure cooker. Same like pressure cooker. Very large size vessel. Okay. Around 2.5 feet height. But the interior structure just like the function of pressure cooker. That means for 15 minutes you are putting the petri dish or test tube inside the air container like things. Here you can put your test tube, your petri dish. You want to sterile all things. You have put here glasswares. All these things are glasswares. Or you can put the nutrient mediums as well. Nutrient medium as well. Okay. Put all these things for 15 minutes. When 15 minutes will over, just a whistle, just like in pressure cooker, a whistle comes out. A whistle will comes out, and here steam releasing chamber is there. Steam will be released with here. Okay. So you can see the functioning is like pressure cooker. So this apparatus is called autoclave. So what is the main theme of this autoclave? High temperature and high pressure. Okay. Now, I have also used here the word chemicals. For chemicals, what will you do, student? You will do filter sterilization, filtration process. Filter sterilization for chemicals you will use. Okay, student. Now, what about the surface? That means you want to do only the surface sterilization. For surface sterilization, you will explant. You will use the explant. What is explant? Student, any part of plant body which you will use in tissue culture, that is explant. So we are reading tissue culture. That is explant. Now, in explant, how will you sterilize? You will give antimicrobial agents. Antimicrobial agents. I'm using the word antimicrobial. Antimicrobial means it can be antibacterial, antifungal, antiprotozoal, because antimicrobial, I'm talking about the full microbes. So what you are using as an antimicrobial agent system? What you are using? We are using ethanol. We are using hydrogen peroxide H2O2. We are using mercuric chloride. Mercuric chloride. And you are using Corox. CHOROX. Corox. Now between all between all these things, which is used as a maximum in lab? Ethanol. Ethanol is used maximum in lab. Is used maximum. Clear with the sterilization purpose, student? Any doubt in sterilization? Any doubt in sterilization? Students?
No, ma'am.
Any doubt in sterilization? Okay, fine. Now we completed with sterilization. Okay. Now, types of tissue culture. Okay. Types of tissue culture. Nutrient media. And the another one is explant. Now in nutrient medium, you want to do tissue culture. So what you will use? You can use callus as well as you can use suspension. What about explants? Protoplast, meristem, and the embroidery. So these are the types of tissue culture. So let us deal in discussion with callus and suspension. I'm starting with callus and suspension. Okay. Starting with callus and suspension. Callus and suspension. So firstly, I'm dealing with callus. Types. Callus. Now, in callus, what do you require, student? You require solid nutrient. Solid nutrient. Medium. Okay. Which medium you will use here, student? I've taught you about one medium, you know, all of you know that medium. I've taught you, I think we have read. What is that medium which are used for bacteria? What is that medium? Tell me. Can you give me one example? That solid nutrient medium is agar. From where this agar is isolated? From where this agar is isolated? Red algae. Can you give me one example of red algae from which agar is isolated? Example of red algae? Example of red algae?
Gracilaria and Gelidium.
Gracilaria. Okay. One example I can write here. Gracilaria. Second example is Gelidium. They are also called agarophytes because they produce agar. Now, hormones are required. What are the hormones which we will take here? In yesterday's class, I have taught you. What are the hormones? Two important hormones which we will use in tissue culture.
Cytokinin and toxin.
Cytokinin and toxin. And we are producing callus, so explant should be larger in size. Explant larger in size. Now, what is the procedure? Let us see. Second one is actually suspension. Okay. That I will discuss later on. Now, see the diagram. Suppose this is the test tube. Okay, in this test tube, what is filled? We are filled with agar. Now, what you will add here? You will add here explant. Suppose this is your explant. This is your explant. Okay. So what is present here? Here medium is there. Agar is the medium. And explant. You have added the explant. Now what you will see here? You will see here dedifferentiation. Dedifferentiation. What is the meaning of dedifferentiation, student? Any one of you can define me dedifferentiation? Any one of you? Dedifferentiation, students? Any one of you?
The dividing cells will be converted into permanent cells.
No. The
No,
reversal of
permanent
this.
cells.
Dedifferentiation means here. Dedifferentiation means when permanent cells shows division. When permanent cells shows division, they are called dedifferentiation. Okay. Now this is the explant. Now explant will show the division. Explant is a permanent cell. Permanent cell means, what do you mean by permanent cell? Where meristematic activity is lost. Now this cell will show dedifferentiation. Okay. So what you will get here, student? This is your medium again. This is your medium. This is your medium. Okay. And this is the callus. Earlier I taught you what is the meaning of callus. Undifferentiated plant growth. Means it is not differentiated into root, stem, leaf. Just unorganized mass, which is showing active division. It is showing active division. Now your callus has been formed. Now this process is slower in comparison with this suspension method. So this is what about the callus method. That means how we are forming the callus. Is this clear?
Ma'am, clear.
Is this clear, students? Callus method clear?
Yes, ma'am.
Next is the suspension method. No? Next is the suspension method. Let's go for suspension method. Second one, that is suspension method. Now in suspension method, students, we are using liquid. Okay, that means when agar is in the liquid form, it is called suspension. Agar is the solid form. Okay, and when you are composing the nutrient medium in a liquid form, They are called suspension. So here you are using the solid nutrient medium. Solid nutrient medium means here you will not use agar. If in comparative way I will say you, so here you will not use agar. Here you will use only oxine, no cytokinin, only oxine. Explant is smaller in size. So just in a comparative manner, I am talking regarding callus culture. Explant is small size. Okay, now let us see the procedure. This is the conical flask. Okay. In this conical flask, liquid is there. This is the liquid. Where is your explant, student? Suppose this is your explant. So explant. And this is suspension. Okay. Now, what will happen? The conical flask. Now, this is the tissue culture. So, these are your suspension. Now, group of cells are formed like this, like this, like this. Okay, what you will get, student? Single cells or group of cells. Now, here what needed, student? Constant stirring is needed. Okay, constant stirring is needed. Okay, constant stirring is needed. Now, stirring about what, student? Around 100 to 250 revolution per minute. In some book, you will also get RPM. Full form of RPM is revolution per minute. 100 to 200 RPM. So what will be the advantage, student, if you are stirring this? Advantage will be that homogeneous mixing. That's why you are rotating it in 100 to 250 revolution per minute. Mixing will be homogeneous. Homogeneous mixing of nutrients. So if homogeneous mixing of nutrients will be there, so what will happen, student, as a result of this? Availability of nutrient will be increased to each cell. Availability of nutrient each cell. It will also increase. If nutrient availability will increase, then cell division will also increase. Okay, cell division will also increase. And in this method, one more thing, student, proper aeration is also required. Aeration means intake of oxygen. You have to provide this culture with oxygen. So in this method, growth rate is faster. Growth rate is faster. So this is about the suspension culture. Clear with it, students? Yes, Devashri, Pranjali, Ritu, Parindita. Clear with this? Avivsha?
Yes, ma'am.
Avivsha, is it clear? Avivsha?
Ma'am.
Yes, tell me. Tell me.
सस्पेंशन मेथड को टेस्ट ट्यूब में करने से भी वही सेम रिजल्ट होगा?
हाँ, but the disadvantage will be that ना टेस्ट ट्यूब में क्या होगा तुम बताओ? टेस्ट ट्यूब में उसको सरफेस कम मिलेगा ना, टेस्ट ट्यूब का दिखने में कैसा होगा, इस तरह का टेस्ट ट्यूब होगा ना, इस तरह से उसको सरफेस एरिया कम मिलेगा। लेकिन कॉनिकल फ्लास्क में क्या होगा, देखो ब्रॉडर बेस है ना, उसको सरफेस एरिया ज्यादा मिलेगा। क्लियर देवश्री?
मैम
Any more questions? Any more questions?
मैम
Now, next, how we will form the plantlets? Okay, how we will form the plantlets? See. Formation of plantlets. Formation of plantlets. One by shoot root regeneration and other by somatic embryo regeneration. Now, shoot root regeneration. What we'll do in this one? This one. Take a test tube. This is the medium. And this is the callus. This is the callus. This is nutrient medium. Okay. Now what will happen? Medium. And here 3 centimeter plant has already. Okay. How much? 3 centimeter short. What will do here, students? 3 centimeter short plants. You will cut the shoot. You will cut the shoot. Okay. Now, next step, what will happen? You cut the shoot, no student. Now, in this step, what will happen? Root will generate. Okay. These are the root actually, student. These are the root. Root will generate. Firstly, root will generate because concentration of oxygen you have increases. And then the shoot generation will take place. Firstly, root generation. Then after root, again, shoot will generate like this. Now, this is your plantlet. You will isolate this plantlet. And now it is ready to go. This is by shoot root regeneration method. It is by shoot root regeneration method. Is it clear? Is it clear?
yes, ma'am.
Now this is by root shoot regeneration. The second method is somatic embryo regeneration. Now what we'll see in this method? Somatic embryo regeneration. Now you have vessel. In vessel you have callus. Increase the concentration of oxygen in the callus. Embryo will form. Now this embryo is formed without fertilization, without sexual reproduction. That is why this embryo is called somatic embryo, or they are called Embryos. Now, what are the stages of embryo development that you have already read in class 11? So, I'm just showing you the stages that you will get in actual embryo development. Firstly, globular stage, then heart shaped, then this shaped, then the final stage like this. So, same embryo development stages you will see in tissue culture as we have seen in the normal embryo development, which are formed with fertilization or through sexual reproduction. Now, what we will do with embryo? With this embryo, now put this embryo in the medium, that means in the test tube. Now, what will happen here? Plumule will form shoot and radicle will form root. So, both root and shoot will form with this embryo. Plumule, with plumule, shoot will generate it. With radicle, root will be generated. So, your plantlet is ready. So, this is somatic embryo regeneration. Clear with this? Now, we have isolated the plantlet. Then the next step, till these steps are clear to everyone. Yes, Ritu, Devashri, Pranjali,
Yes, ma'am.
Paringita,
Yes, ma'am.
till these steps are clear. So we have formed the plantlets. Tomorrow is last class for this strategies for enhancement. Huh? From Monday, I will start human health and disease. So we have isolated the plantlets. The next step after plantlets is hardening. What is the meaning of this hardening? So we have removed the plantlets. Plantlets are removed from culture vessels. Now, with culture vessels, it is shifted to earthen pots. With earthen pots, they are shifted to mist benches. Now, what is this mist benches? These are the apparatus where you will get high humidity, light intensity decreases, and you will keep in this mist benches for 3 to 4 weeks. Now, what is the condition, student? Try to understand the phenomena. Humidity is high and light intensity is less. So we can say now that we have kept this plantlet under stress condition. We can say that we have kept the plants in stress condition. Now, under stress condition, a plant is producing a hormone. What is the name of that hormone? What is the name of that hormone? Stress hormone. In plant, which is called stress hormone?
Abscisic acid.
Abscisic acid.
Abscisic acid. Now, the plant is ready to get the stress condition, that is, it can tolerate the high humidity, it can tolerate the low light intensity. Now it is ready. Now you give it to farmers. Now it is ready for farmers. Clear with the two type of culture, suspension culture and canvas culture. Clear with this? So our next type is protoplast culture. Clear with this?
Yes.
Clear with this? So our next is protoplast. Protoplast culture is also called somatic hybridization or parasexual hybridization. These are the name: somatic hybridization, parasexual hybridization. What is it, student? Process of fusion of. Firstly, write down the definition. Process of fusion of genetically different cells. Okay, here we are taking genetically different cells. Genetically different cells means here you will form hybrid cells. But these hybrids are somatic hybrids. So we will form hybrid cells with somatic body. With somatic bodies, these are called hybrid plantlet. These are called hybrid plantlet, or they are called somatic hybrid. Okay. Now you see the steps. Next, explant which we are choosing here, they are thin cells. Now it can be from leaf where, student? From leaf mesophyll cells. You can take, or you can take from stem cortex, that is cortex of the stem. In anatomy, we have seen the cortex region of the stem as well as root. So you can take from the cortex of the stem. Now we'll see the techniques. What are the techniques? Suppose what are the techniques, students? See the techniques. Plant A, and this is plant B. You are treating the plant with cellulases because it will break down. It will break cellulose or pectinases. It will break down. You have to break down of pectinase and cellulose. They will digest the cell wall. Now, if cell wall is digested, they will digest the cell wall. If cell wall is digested, the structure which is formed, they are called protoplast. They are called protoplast. This is one structure. This is the nucleus. Cell wall is degraded. It is the nucleus. There is a plant A. It's for plant A. The second one. It's the nucleus. This is for plant B. And these are the cytoplasm. And these are the cytoplasm. Okay. Now fusion. Fusion with the help of, what are the chemicals which we will use here? We will use here PEG. Full form of PEG is, yes, PEG, PVG, sodium nitrate. These chemicals you will use for fusion. Now, full form of PEG is polyethylene glycol. PVG, polyvinyl glycol. These are used for fusion. Okay. So fusion method can be chromofusion with the help of chemicals or electrofusions. That means fast electric field with this. Now you will get hybrid cells. You will get hybrid cells. Now the hybrid cells which you will get, that contains cell wall. Cell wall is present. So how will you get this? The basic callus. From callus collect the plantlet, then produce somatic hybrid. This is the basic. Now what kind of hybrid can be formed? Three types of hybrid can be, that I'll discuss later on. First you tell me, somatic hybridization or parasexual hybridization or protoplast culture is clear to you or not? Is this clear to you or not? Tell me.
Yes, ma'am.
Ritu. Varindita, Pranjali, is it clear? Is it clear?
Yes, ma'am.
Any doubt? Any doubt with this? बोलो देवाशी.
I'm clear.
Now, suppose this is the callus. The cell which we are forming, this hybrid. Now, it contains the nucleus of parent A as well as the nucleus of parent B. That is not changed. Wait. It contains the nucleus of parent A as well as it contains the nucleus of parent B. Okay. Cytoplasm of parent A as well as the cytoplasm of parent B. Now, this type of cell, this type of hybrid is called symmetrical hybrid. Both nucleus and cytoplasm equally distributed. That is the symmetrical hybrid. Second one, asymmetrical hybrid. This is the nucleus of parent A. Cytoplasm from both the parents. Okay, cytoplasm from both the parents equally distributed. But you can see the nucleus distribution is asymmetrical. So this type of hybrid is called asymmetrical hybrid. Okay. Third type. This is the cell. Nucleus of only one parent. Only one parent. That is parent A. But cytoplasm of both the parents. Cytoplasm A. Cytoplasm B. This type of hybrid is called cytoplasmic hybrid or cybrid. Cytoplasmic cybrid. Cybrid. Clear with the types of hybrid produced with this callus? Symmetrical hybrid, asymmetrical hybrid, and cytoplasmic hybrid. Clear with this? Tell me. Clear with this? Yes, Parindita, the Bashri is clear with this. Any doubt?
मैम साइटोप्लाज्मिक हाइब्रिड में न्यूक्लियस टोटली एब्सेंट रहता है पूरा.
No, no exchange of nucleus. Only one parent, either from parent A or from parent B. So are drawn from parent A. Okay, that is why it is called cytoplasmic hybrid. That means both cytoplasm. Cytoplasm will come from both the parents, but nucleus only from one parent. Clear with this, Devashri?
Yes.
Now, Carlson, very important. The scientist, he formed first to form hybrid in tobacco plant. In glaucoma. Wait a minute, students. Wait a minute. He formed tomato. Tomato means the hybrid of potato and tomato. But this tomato is not commercially viable. Not commercially viable. Clear with this? So we have done with our today's class. Yes, Devashrit, Parindita, any doubt from the whole lecture? Any doubt from the whole lecture? Devashrit, Parindita, Anjali, Ritu, any doubt? Ashwini, any doubt? Ask me. Any doubt you can ask me? Students? Okay, tomorrow is our last class for tissue culture. Okay. Thank you, all of you.